Hello everyone, welcome back to my channel. My today's topic is cell membrane transport. So this covers cell membrane as well as fluid mosaic model, and about cell already I have taken in my class. So first, before entering into cell membrane transport, I just want to recap what is meant by cell. A cell is a structural and functional unit of each and every living organism. So this is a person who has proposed cell, Robert Hooke, in 1665. So in this topic, there are some terms you should know. Concentration, solute, solution, concentration gradient. So concentration is amount of the solute in a solution, and the solute which dissolved substance in a solution, and the solution a mixture of both. Concentration gradient, the gradual difference in the concentration of solutes in a solution between two regions. So before entering into the topic, we have to take care of plasma membranes. Why should we have to take care? Problems with the cell membrane are involved in many diseases, such as type 2 diabetes, organ transplant rejection, cholera, and cystic fibrosis, also cancer. So first, membrane transport, it is a boundary between inside and outside of the organ. So it can be impenetrable boundary. Here, whatever the food we are taking inside, carbohydrate, protein, lipids, whatever it may be, amino acids, lipids, etc., as well as particles, waste, ammonia, salts, carbon dioxide, water products, it has to be existed. So in and out takes place with the help of plasma membrane. Now this is the structure of plasma membrane, very important 5-mark question for university exam, which is called as fluid mosaic model. Here Davison and Daniel originally proposed membrane structure in 1935. Singer and Nicholson in 1972 described the structure of biomembrane as a fluid mosaic model. So here the structure shows clearly it has a phospholipid bilayer. Phospholipid bilayer, 90% of it covers in the cell membrane or plasma membrane. It has hydrophilic head and hydrophobic tail. So hydrophilic head attracts the particles, whatever we are taking inside. With the help of tail, from ICF to ECF or ECF to ICF, it will be transporting. Here the integral proteins as well as extrinsic proteins or glycoproteins, in which proteins attached with the sugar molecules. So you have to name the parts also. When you're drawing the diagram with color pens, you have to use the parts with phospholipids, integral protein, peripheral protein, glycoproteins, and very important thing which I have not mentioned here is cholesterol. Cholesterol in between the plasma membranes, it is present in order to maintain the plasma membrane fluidity. So next, composition of the cell membrane, it contains lipids, which is called phospholipids, sterols, and proteins, integral, peripheral, carbohydrates, glycolipids, and glycoproteins. So these are all keywords we'll be expecting when we are correcting the papers. Not only drawing the fluid mosaic model is enough to get full marks, but you have to differentiate the structure with parts as well as functions. So plasma membrane is metabolically very active and highly selective. That is very important keyword. It is permeable, whatever, for nutrients, so that entry and exit of the compounds are well regulated. Membranes are mainly made up of lipids, proteins, and small amount of carbohydrates such as glycoproteins and glycolipids. Here peripheral proteins loosely attached by ionic bonds. Example, cytochrome C of mitochondria. And integral proteins, they are deeply rooted by hydrophobic bonds. Example, channels and receptors. So these are all the key terms you should mention in fluid mosaic model as a function. Lipids, as already I have told, phospholipids, hydrophilic head, hydrophobic tail. A small diagram is enough to give a good presentation, which is amphipathic. And cholesterol alters the fluidity of the membrane present in between the phospholipids. Increased cholesterol, less fluidity on outer surface and more fluid in the hydrophobic core. So fatty acids, cis form increases the fluidity, Transform decreases the fluidity. For MCQ, they may ask, what is the function of cis and trans? Carbohydrates 2 to 10% contributes by the sugar residues of glycolipids and glycoproteins. So commonly, as a summarization, function of cell membrane I have given. Cell membrane separates the components of a cell from its environment, surrounds a cell. It is a gatekeeper of the cell that regulates the flow of materials in and out of the cell, which is called as selectively permeable. Cell membrane helps to maintain homeostasis, stable internal balance, and also it keeps a proper concentration of nutrients as well as water and eliminates waste particles. So next, transport. Now we are entering into detailed question. Transport of molecules across membrane. It may be active or passive. Mostly active transport will be asked in 5-mark question, but in detail if they are asking, you have to write both passive as well as active. So first we'll see what is meant by active transport. The picture shows a person standing up the hill. So there is no need of energy to come down, whereas there is need of energy to climb up the hill. Here passive transport, a solute molecules from high concentration to low concentration across a membrane. So this shows, this clear picture also can be clearly mentioned in this answer, because high concentration to low concentration, a keyword we'll be expecting in this point. So it is a downhill transport and requires no energy. It is classified as simple diffusion and facilitated diffusion. So for simple diffusion, a picture, a diagrammatic thing I have shown here, it moves from high concentration to low concentration in order to maintain homeostasis. So passive transport, no energy required through the channel protein and lipid bilayer. Example, transport of oxygen, all gases, carbon dioxide, nitrogen, ethanol, and urea. So diffusion is nothing but a movement of small particles across a selectively permeable membrane until equilibrium is reached. Next, facilitated diffusion is a movement of larger molecules like glucose through the cell membrane. Larger molecules must be helped. So in this case, proteins in the cell membrane form channels for larger molecules to pass through are called protein channels. For example, I have given ping pong mechanism. This is very important facilitated diffusion question as a subdivision marks or 2 marks question. So when we are asking for 2 marks, this slide is more than enough to give 2 marks for you. On subdivision, sometimes they may ask. In that case, you have to draw this diagram. Ping pong mechanism. Actually, pong state comes first. It is opening towards the concentration, whereas here ping state, it is opening towards lesser concentration. So in that case, it shows higher solute concentrations accepted by protein molecules, and it will be traversed from intracellular fluid or extracellular fluid to another site, that is called ping state. So ping pong mechanism should be there for 5-mark question or whatever. Facilitated, if they are asking, kindly mention this diagram. This is very important. So next, osmosis, 2-mark question, which comes under diffusion. It is a diffusion of water through a selectively permeable membrane as that of cell membrane. So it is semi-permeable, which is permeable to water but not to sugar. So water diffuses across a membrane from an area of high concentration to area of low concentration. And these are all the terms you have come across in physiology classes, I think. Hypertonic, hypotonic, and isotonic. So no need to revise again, but you should know when the cell edema happens. So when the balance will be turned into imbalance. So hypertonic, hypotonic, and isotonic should be there. Next comes very important 5-mark question, active transport. So it is a movement of molecules from low concentration to higher that needs a protein pumps. Example, body cells must pump carbon dioxide into the surrounding blood vessels to be carried to the lungs for exhalation. So next, active transport. 2 types are there. It needs external energy, ATP molecules, whereas in passive transport, no energy requirement is there. Here, energy requirement is there. There are 2 types: primary active transport, secondary active transport. Here, energy is derived directly from hydrolysis of ATP, whereas secondary, it provides the energy for transport indirectly. With the help of primary active transport, secondary will be running. So that's why it is called a secondary active transport. When we are asking active transport, you have to mention both primary as well as secondary. So first, this is primary active transport. Diagram is very important. With the help of diagram only, you have to explain this. So primary active transport, example, sodium, potassium, ATPase. It maintains intracellular cations and thus a in cell volume. So protein synthesis by maintaining high concentration of potassium. This is very important. They'll be asking for subdivision question. Why ICF potassium is high when compared to ECF? In order to maintain the metabolism of carbohydrates, lipids, proteins, potassium is helping a lot. That's why ICF potassium should be high. So this is a diagram for 3 sodium molecules, 2 potassium. 2 potassium will be coming inside and 3 sodium will be going outside. With the help of proton pumps, ATP energy is required here. This is extracellular space and intracellular. You have to mention these. Some students will be just drawing the solute particles and they'll not indicate whether it is a sodium or potassium. So just mention the solutes. What are the solutes are transported with the help of ATP molecules. That gives a good presentation with full marks. So next, this is additional calcium ATPase as well as hydrogen potassium ATPase. If you want to get full mark, you can copy this because this is for muscle contraction, HCl secretion, urine acidification. So this is extra, but if you want, you can write it. This next comes secondary active transport. Transport of one solute against its concentration gradient by using the energy generated by gradient of another solute transporter. So example, reabsorption of glucose from kidney tubule or intestine. So with the help of sodium potassium pump, along with the sodium sugar molecules also will be transported. So with the help of primary, secondary will be acting. That's why it is called as secondary active transport or sodium co-transport. So next, this is additional thing. Very important, endocytosis and exocytosis. Mostly they'll be asking as a question phagocytosis in this case. So phagocytosis, you will be telling cell drinking, cell eating. So phagocytosis, engulfing of food particles. Here also we're going to see endo as well as exo mechanism by which very large molecules. For active and passive transport, small molecules, endo and exo for large molecules such as food and waste get into as well as out of the cell. Here WBC, which are part of the immune system, surrounds and engulfs bacteria by endocytosis. So diagram is very important. How far the food particles entering inside and how the waste existing out. So this is very important question also. When compared to active passive transport, sometimes they may ask this question also. So next, food is moved. I have explained with the diagram. So side by side like this you can present. With the diagram you can explain as a point. And this is summarization of transport of larger molecules, phagocytosis, pinocytosis, and receptor-mediated endocytosis. So phagocytosis is nothing but cell eating. Pinocytosis, cell drinking, and with the help of receptors, best example you can take insulin receptors. So with the help of insulin receptor, insulin uptake of glucose. So that is a transport of larger molecules, and I've shown a small summary recap of uniport, symport and antiport. Most of the students will be writing this slide when they are asking about active and passive transport. So this is example we have given for active and passive transport and not fully covered for active. Listen, so this slide just show an example for uniport. Single molecule will be transporting, whereas symport, two molecules same direction, whereas antiport, two different in different directions, two different solutes. So you can tell example, simple diffusion, so symport sodium co-transport, and this is primary active transport. So like that you have to give example alone and not mention for full 5-mark question. So uniport I have given example, transport of glucose into RBC, symport sodium and glucose, antiport exchange of chloride and bicarbonate as well as sodium potassium transport. So these are all examples I have given again and again because this is very important question. So this is overall summary of passive transport and active transport. All together I have given concentration gradient, carrier protein and protein channel protein, as well as with the help of lipid bilayer I have given. So this diagram also gives